Unit 12. So the first part of Unit 12 is talking about ubiquitin. So the first thing I know is the ubiquitin conjugation story. It's on a slide. It looks like this. It tells the whole thing about enzyme 1, 2, and 3 and what they do. You could ask a couple questions about that. So I'd be familiar with that. The only thing to know about proteasomes is that they are the ones that actually degrade. It's the executioner of proteins, as the slide says. Might be a question about the binding site being the N-terminal threonine nucleophile. That's the first part of the unit. Pretty simple. Not a lot of detailed questions come off that. And the next part about amino acid degradation should be a review. So to do a quick review, you have the amino acid and alpha-ketoglutarate react together. That's transamination, done by transaminase or aminotransferase, same type of enzymes. That's B6, pyroxene, or PLP dependent. And then your product is glutamate, and that's going to react with NAD plus and water to form back the alpha-ketoglutarate, NADH plus NH4 plus, which is going to go to the ammonia is going to go to the urea cycle. That's what the whole unit is going to be about at the end. The second reaction is the oxidative deanimation, which is done by glutamate dehydrogenase. Dehydrogenase, niacin dependent, B3. Both of those enzymes are found in both the cytosol and the mitochondria, either or. And remember, this is reversible too, so you're going to see some synthesis of amino acids, and in that case the arrows would just be drawn the other way. So both of these reactions are totally reversible. If the arrows are coming down here and going up, it's going to be degradation. If the arrows are starting here, you're synthesizing an amino acid if the arrows are going the other way. Same reactions happen both ways. Two exceptions to how this works is serine and threonine. They each have their own enzymes. They can be directly deanimated. So serine will go to pyruvate and ammonia. Threonine will go to alpha-ketobutyrate and ammonia, which will both go to the urea cycle after that. And those are done by dehydratases. Serine dehydratase and threonine dehydratase. So the next unit is what happens if you break down an amino acid in the peripheral tissue, say in the muscles. The liver is going to do all the processing. So first we need to get it to the liver. And you're going to transport it by two amino acids. It's either going to be alanine, and on the next piece of paper we're going to look at glutamine as the other one. They each, that process is different for both of the amino acids. So where this starts out, this looks the same as what we just saw on the last page. You have this broken down amino acid, which we're going to start right here. The amino part of it is going to react with these guys and alpha-ketoglutarate through the same reaction backwards. Then you're going to have glutamate, you'll have these other products here. Pyruvate's going to come in out of nowhere. Those are going to react together from your aminotransferase reaction, and you're going to have alanine. So alanine is a happy amino acid to be sent from the peripheral tissue through the blood to the liver, and then you'll get rid of it through the urea cycle. Urea cycle is being used for the amino group, by the way, and the carbon skeleton of it will be used for gluconeogenesis. The body is very efficient. And once it gets to the liver, everything is just reversed. So alanine will go to the liver. So if we're going back to this first thing, alanine, now we're in the liver, it's going to react with alpha-ketoglutarate and eventually becomes urea to the urea cycle. So alanine can be an example of what happens here. Remember, if it's alanine, it's always pyruvate as the alpha-keto acid. And that's the part that would go to gluconeogenesis. So alanine is pretty simple. It's using the same format as we've seen before. Glutamine does something different, totally different. First, you're going to make the glutamine by taking the amino group glutamate and ATP, changing it to glutamine here. That's done by glutamine synthetase. It's using ATP synthetase, so it's a ligase, class 6. So now this glutamine is going to go through the blood to the liver, and it arrives in the liver where it's going to react with water and change back into glutamate and ammonium, which is going to go to the urea cycle. The reaction, the enzyme that does this reaction is glutaminase, which is a class 3 reaction. It's a hydrolase. That's a liver mitochondria enzyme. That's one of his favorites to ask about. So this glutamate is going to come down here and react with oxaloacetate coming out of nowhere. Glutamate plus oxaloacetate gives you alpha-ketoglutarate and aspartate. Aspartate is a key amino acid in the urea cycle, so both of these products are going off to the urea cycle, which we will talk about now. So I think this is the last major pathway that you have to memorize in Biochem 2. So be happy about that. The first reaction, you have HCO3 plus 2 ATPs plus the ammonium. So remember, this is off to the urea cycle. Here it is. And that's going to be done by carbamyl phosphate synthetase 1, abbreviated CPS1. It's a synthetase, so it's a ligase, class 6, and it's found in the mitochondria. As far as location, these first two are going to be mitochondria, and the last 3 are going to be cytosol. So now you have this carbamyl phosphate and these byproducts, which no one really cares about. So the carbamyl phosphate is going to come down to the next reaction and react with ornithine, if I'm pronouncing that correctly. OTC is the enzyme for this reaction. It's transferring the phosphate, so it's a transferase or enzyme class 2. And the product is citrulline. I think this stands for something like ornithine transcarbamyl something. You'll have it in your notes. OTC will be the abbreviation. So the citrulline we're going to bring down to the next step here. And that's going to react with aspartate. Remember on the last one we had this aspartate coming. Here's one source of it. So this aspartate will end up right here. So we have another synthetase, meaning we have another ligase. ATP is being used. So citrulline plus the aspartate plus ATP will give you argininosuccinate plus this other stuff nobody cares about. That's going to react with the enzyme argininosuccinase to give you arginine and fumarate. So a couple things that could be asked here. First, we have a 1 to 2 product, so that's an indicator of a lyase. And fumarate is one of the byproducts here, so that's a Krebs cycle link. If he asks, which enzyme produces a metabolite of the Krebs cycle, or which amino acid is paired with a metabolite of the Krebs cycle. This is the reaction he's talking about. The arginine is what's going to react in the last one. We have a hydrolase reacting with water to form the ornithine, plus the whole point of the urea cycle is to make urea. Hydrolase class 3. And then this ornithine, if we were to draw this out in circular form, this ornithine would be ending up here again. So it's a recycled metabolite. I mentioned the enzyme classes. It's one of his favorite things to ask about. So notice there are 2 enzyme class 6s, 2, 3, and a 4. The ones we did not say there are no redox ones, no isomerases. So if you list 4 of them, if you see a class 1 or class 5, that's not the answer. Regulation is very simple. The key enzyme is that first one on the list here, CPS1, which is stimulated by N-acetylglutamate. We haven't talked about N-acetylglutamate. That's made from acetyl-CoA plus glutamate. That gives you N-acetylglutamate. The enzyme for that is N-acetylglutamate synthase, and arginine is the amino acid that stimulates that. So arginine indirectly stimulates CPS1 because it's making the main activator of CPS1. Arginine, not aspartate. They look the same, don't get them mixed up. Aspartate's the other one in the Krebs cycle but has no role in regulation. Clinical is pretty easy. Any problem with any of the enzymes, you're not going to excrete urea, so you're going to have too much of it built up in the blood, or too much ammonium built up in the blood, not enough urea. Too much NH4+. A couple of the enzymes that could be a problem would be argininosuccinase, which in that case you're going to supply lots of arginine to try to take care of that. And if you have problems with not having enough CPS1 or OTC, you're going to supply an excess of benzoate or phenylacetate. That's Unit 12.